Oncogenesis and Metabolism of Xenobiotics Part 4. Today's lecture is focused on studying the pharmacological, toxicological, immunological, and carcinogenic responses to xenobiotics. Then we'll see the differences between oncogenes and proto-oncogenes. We'll study in detail mechanism of oncogenesis. Then we'll take concept of tumor suppressor genes. And lastly, we'll deal with role of oncogenic viruses in the promotion of cancer. First, we are going to see pharmacological responses which are associated with xenobiotics. If a xenobiotic is a drug, then it is mostly available in its prodrug form, that is its inactive form. Now, when prodrug undergoes phase 1 reactions of xenobiotic metabolism, then this drug may be converted into its active form if it is beneficial for the body. But if the drug is already available in its pharmacologically active form and it is not needed by the body or it is harmful for the body, then through phase 1 reactions, its action may be terminated or ended. There is a very diverse class of science which is called pharmacology, in which we study the diverse effects which are produced by these drugs. Responses that are generated against drugs are always different in every individual. This is because that everyone has its own unique or specific genetic material or genome. Today we are going to see 4 important reactions or responses which are generated against drugs, and these responses are always different in every individual just because of the alteration in the enzyme and protein structure in that specific individual. There is another big domain of science which is called pharmacogenetics that basically studies the effect of the genetic material on the response which is generated against the drug. In this column, we are going to see the enzyme or protein affected in one specific individual, and then we are going to see the reaction, response, or consequence of this altered genetic material on the drug. For example, if an individual is having mutations in glucose 6-phosphate dehydrogenase enzyme, in HMP pathway, we have discussed this mutation in detail. Then if the same person is going to ingest primaquine, then that person would be having hemolytic anemia. This hemolytic anemia is basically a response or the reaction which is associated with the effect of this drug on this glucose 6-phosphate dehydrogenase mutation. So basically this drug does not produce hemolytic anemia if there is no glucose 6-phosphate dehydrogenase mutation. But if glucose 6-phosphate dehydrogenase is mutated in any individual, and if the same individual is going to ingest this drug, then this will be the consequence, that is hemolytic anemia. Similarly, if anyone has a defect in calcium release channel, which is also called as ryanodine receptor, in the sarcoplasmic reticulum, then as a result of which, if the same individual is administered anesthetic, for example halothane, then he or she would be developing malignant hyperthermia. So this effect is associated with the mutations in the genetic material in the genome, and as a result of which this drug is showing this effect, that is malignant hyperthermia. extreme production of heat in the body that may lead to fever and ultimately death of the individual. Then similarly, if there is mutation or polymorphisms in CYP2D6, and if the same individual is going to take depresoquine drug, then there will be the slow metabolism of this drug in that individual. And as a result of which this drug may be accumulated and may result into some unwanted effects. Similarly, if there is mutation or polymorphism in CYP2A6, as a result of which the effect of nicotine in that individual would be different. There would be impaired metabolism of nicotine in that individual. And nicotine, it would be available in its active form in that individual. It won't be converted into its inactive form. So therefore, that person can become tobacco independent smoker. Or he may get protection to become a tobacco dependent smoker. Now we are going to see toxic effects associated with xenobiotics. There are some xenobiotics which have extremely high toxicity. And even if they are taken in very minute amounts, they may be proven very toxic and very harmful for the body. For example, cyanide. And even on the same hand, there are some drugs which are, even if taken in large amounts, cannot be very much toxic for the body. So the effects associated with xenobiotics are varied, are wide. So today we are going to see only the major effects or the key effects which are associated with the effect of toxic xenobiotic on the body. For example, toxic xenobiotics can impart cellular injury, which is called cytotoxicity. Then toxic xenobiotics may bind to a protein, for example, any immunological protein, and as a result of which they may change the antigenicity of that protein that we are going to see with the example on the next slide. And then there are certain toxic xenobiotics that may trigger development of cancer, because these toxic xenobiotics may act as carcinogens. So this slide is explaining that xenobiotic is metabolized into reactive metabolite. And if this reactive metabolite is not converted into non-toxic metabolite by some protective mechanisms, then this reactive metabolite may impart cytotoxicity. It may result into immunologic damage, resulting to again cellular injury, and it may also result into carcinogenesis, that is development of cancer. Normally what happens that whenever xenobiotic is exposed to body, then it is processed through cytochrome P450 system into a reactive metabolite. For example, it may be converted into epoxide. Then with the help of epoxide hydrolase, it is converted into non-toxic metabolite. For example, dihydrodiol. So dihydrodiol is a non-toxic metabolite which is generated by epoxide through epoxide hydrolase. Now in this situation, this non-toxic metabolite cannot impart cellular injury or it cannot result into development of cancer, and it is simply gets excreted out of the body. Similarly, there are also some other xenobiotic metabolizing enzymes as well, for example GSHS transferase, and this enzyme may also convert many reactive metabolites into non-toxic metabolites. So supposedly if GSH transferase or epoxide hydrolase not available, means these are if defective, mutated or missing, or even any other enzyme of phase 2 is missing, then reactive metabolites cannot be converted into their non-toxic forms. So these reactive metabolites would all be available to trigger cellular injury. How? These reactive metabolites may trigger heptin. Heptin is any small molecule which can trigger antibody production, and these antibodies can target their own cells like autoimmunity and may result into, again, cellular injury. Similarly, these reactive metabolites may cause mutations in genetic material, and as a result of which may induce cancer development. Okay, the second part of today's lecture and the last part of this whole topic is oncogenesis, that is the development of cancer. You can do it from Harper, 29th edition, chapter number 55. Cancer. Cancer is basically uncontrolled growth of cells, and these cells are capable to metastasize and invade, that they can spread the surrounding tissues and even distant tissues. Cancer is ranked as the second most commonest cause of death worldwide. And there are 5 major organs that have been studied to be involved much in the development of cancer. And these 5 organs are lungs, colon, breast, prostate, and uterus. There are 3 main properties of cancer cells. Number first, they have high cellular division rates, means that they can undergo multiplication, very fast multiplication. They have increased or high mitotic rates. They can undergo immense proliferation. Then the second capability of cancer cell is to invade or spread to local tissues. And then in its advanced stages, it may metastasize. It can spread to even distant organs or tissues of the body as well. 6 major features of cancer cells. Cancer cells are capable enough to invade to local tissues, to local surrounding tissues. Even cancer cells can metastasize. They can spread to distant organs and tissues as well. Then cancer cells are self-sufficient in their own growth signals. They don't rely on other cells to produce growth signals for cancer cells. Cancer cells can produce their own growth signals to promote fast multiplication. Then cancer cells are insensitive to anti-growth signals. If body or host is trying to kill cancer cell to inhibit its growth, then there would be no effect on the growth of cancer cell because cancer cells are insensitive to anti-growth signals. The fourth most important feature of cancer cell that cancer cell has its own angiogenesis potential, which means that cancer cell has its own sustained growth of blood vasculature or blood vessels. It can supply blood through vessels to all its cells so that nutrition can be provided to cells every time. Then the cancer cells have limitless replicative potential. That means that they can replicate beyond limits. There is no end to the replication of cancer cell. So therefore, cancer cell is considered as immortal. It can divide, divide, and divide. And another feature of cancer cell that there is no apoptosis or programmed cell death of cancer cell. Therefore, cancer cell can survive for longer period of time. So now we are going to look into causes of cancer or etiology of cancer, then the effect of carcinogenic agents on the carcinogenesis. Then these are simply called as carcinogens, that is cancer reducing agents, or mutagens that can cause mutations in the genetic material, and as a result of which cancer may be caused. So there are three main causes of cancer. First is physical, that is via radiant energy. Then we have the cancer induction through chemicals, and these chemicals are called chemical carcinogens, and the process is called chemical carcinogenesis. And then we have biological cause of cancer, and that is the tumor viruses, mostly the retroviruses or oncogenic viruses, which may induce cancer. So radiant energy, chemical carcinogens, and certain viruses are the main cancer causing agents. First cause of cancer is physical, which is induced by radiant energy in the form of X-rays, gamma rays, or UV rays, which are mutagenic or carcinogenic. These rays can damage DNA by imparting some direct effects or by some indirect effects. This is the list of direct effects on the DNA which can result into impairment of DNA. First, these rays can cause breaks in the single and double strands of DNA. These rays may result into elimination of nucleotide bases. They may impair the process of cross-linkage of DNA strands, and they may also result in the formation of pyrimidine dimers. In spite of purine pyrimidine dimer, pyrimidine dimer can be formed, which can destabilize DNA and ultimately may cause cancer. All of these basically are due to result of spontaneous mutations, those mutations that are abrupt, which can take place in no time. Then in the indirect effects, first these rays produce free radicals, or for example reactive oxygen species like hydroxylines, superoxidines, peroxides. Ultimately they all result into oxidative stress and damage DNA, and eventually DNA becomes impaired and it may result into cancer. So UV rays are mostly associated with the pyrimidine dimers formation, and as a result of which DNA becomes damaged. Similarly, ionizing radiations like alpha particles and neutrons, these are also extremely carcinogenic and may involve in the pathogenesis of leukemias and even lung cancer. Then the second most important cause of cancer is chemicals, or chemical induced cancer, or chemical carcinogenesis, or chemical oncogenesis. So drugs, toxins, pollutants, even many other xenobiotics, all may act as chemicals and may result into cancer development. For example, polycyclic aromatic hydrocarbons, or PAHs, for example benzopyrene, these chemicals are involved in the development of lung cancer. Then azodides are involved in the development of blood cancers. Nitrosamines, for example dimethyl nitrosamine, is associated with gastric cancer. Then aflatoxin, for example aflatoxic B1, which is found in mushroom, and it is a type of a naturally occurring compound, it may induce liver cancer. Then certain drugs, for example cyclophosphamides and many others, may also result into different types of cancer. And then there are some inorganic or synthetic agents, for example arsenic, asbestos, nickel, chromium, and many other metals, which can also result into cancer development. Chemical carcinogenesis is the cancer which is induced by chemicals. There are two stages which are involved in chemical carcinogenesis. First is initiation and the second one is promotion. In the first stage, there is simple exposure of a chemical, and as a result of which there is irreversible DNA damage. And then there is promotion, in which that initiated cell can undergo growth and proliferation. So initiation and promotion can result into the production of abnormal mass of cells, which may then further be categorized as benign or malignant, and this is called as neoplasm. The third most important cause of cancer is biological. That includes oncogenic viruses. Oncogenic viruses are cancer-inducing viruses, and their process of inducing cancer is called viral oncogenesis. 15% of human cancers have been observed to be caused by viruses, and in the viral oncogenesis, both DNA and RNA-based viruses are involved. Basically, what happens that the genetic material or genome of viruses, it becomes incorporated into the host genome. And this process of insertion of genetic material of virus into host genome is called insertional mutagenesis. Now we see the same mechanism in detail. Insertional mutagenesis is mostly caused by RNA viruses, which are also called retroviruses. So the genome of the RNA virus, it gets incorporated into the host genome after reverse transcription. First, there is the use of viral reverse transcriptase enzyme that converts viral RNA into viral DNA, which is called as cDNA. And then that viral DNA is integrated with the host DNA, and as a result of which it causes deregulation of cell cycle, that there is no control over cell cycle and there is massive production of cancer cells. Then inhibition of apoptosis, programmed cell death becomes inhibited and there is no death of cancer cell. Then it may also cause abnormalities in cell signaling pathways, and as a result of which cell signaling pathways become switched on and there is no stoppage in their activity. And there is another important point for you, that there are some RNA viruses which may contain oncogenes already in them. Now what are oncogenes? Oncogenes are cancer causing genes. For example, Rous sarcoma virus, which contains oncogene called SRC. And there is also another virus which is called murine sarcoma virus, which contains oncogene RAS. So SRC and RAS are oncogenes which are present in these viruses, and they may cause cancer directly. And these viruses are simply called as oncoviruses, retroviruses, or oncogenic viruses. Now we are going to see role of DNA viruses in causing cancer. A mode of action of DNA virus is to suppress or to inhibit tumor suppressor genes. Tumor suppressor genes are those genes which normally suppress tumor development in our body. But DNA viruses themselves inhibit tumor suppressor genes, and as a result of which tumor cannot be suppressed in the body and there is more chance of development of cancer. These are the types that we are going to see later on. So here is the list of the viruses, and then their genome is given and then the cancer associated with them. For example, Epstein-Barr virus, which is a DNA-based virus. This is involved in the pathogenesis of lymphoma, for example, Burkitt's lymphoma. Also, it results into the induction of nasopharyngeal cancers and also lymphomas of B-cell based. Then we have hepatitis B virus, which is DNA-based, and this is involved in hepatocellular carcinoma, that is the liver cancer. Then hepatitis C, it is RNA-based virus. It is also the main culprit behind a liver cancer induction. Then human herpes virus type 1 is DNA-based virus, and it causes Kaposi sarcoma, which is mainly seen even in the patients of AIDS, in which purplish spots are seen on the feet and legs of patients. And this cancer may spread from dermis to lymph nodes and even to internal organs as well. Then we have human papilloma virus, and there are many other types of human papilloma viruses as well. And these viruses are DNA based and they may cause cancer of the cervix. Then we have human T-cell leukemia virus type 1. This is RNA based and this is involved in the pathogenesis of adult T-cell leukemia. So oncogenes and their role in cancer development, that is carcinogenesis. Now what is oncogene? Oncogene is basically cancer causing gene. And this oncogene is derived from proto-oncogene. Now what is proto-oncogene? Proto-oncogene is a normal gene that controls growth of the cells, that basically encodes growth stimulating proteins. So proto-oncogene, when it becomes mutated, it becomes oncogene. So oncogene can be called as altered or modified form of proto-oncogene. And this oncogene, it can encode variety of proteins which may affect cellular growth and even cell death program. So proto-oncogenes are the normal genes which are converted into cancer-causing genes called oncogenes whenever proto-oncogenes undergo mutations. You can also see the same concept with this statement that oncogenes are derived from activation of proto-oncogenes. So here you have proto-oncogenes, and if there are mutations in proto-oncogenes, for example, we have seen the physical cause and chemical cause and biological cause, and these three causes can convert proto-oncogenes into oncogenes, and as a result of which there is increased growth of cells which may lead to cancer. On the other hand, we have tumor suppressor genes which control or suppress the growth of tumor in our body. But if tumor suppressor genes are themselves inactivated, when these are themselves inhibited, for example, by DNA viruses, then as a result of which they may also result into increased growth rate of cells, ultimately leading to cancer. Now we are going to see mechanisms that may result into activation of proto-oncogenes into oncogenes. The very first is a mutation. Mutation, for example, point mutation, and this point mutation may lead to cancer. For example, if point mutation takes place in the RAS oncogene, then it may result into increased stimulation of adenylyl cyclase enzyme. And there is abnormality in the cell signaling pathway, and there is abnormal activity of this pathway which may result into cellular proliferation, ultimately leading to cancer. The second most important mechanism is called promoter insertion, in which there is the insertion of viral promoter into the host genome. Now, what is viral promoter? This is a specific region in the virus genome that may promote the synthesis of messenger RNA for making some specific proteins, for example, growth proteins. Now, what is the mode of action of this process? Whenever there is insertion of viral promoter into the host genome, then it activates a gene in the host which is called MYC gene. This MYC gene is another transcription factor which encode many growth proteins, and as a result of which there is cellular proliferation leading to cancer. Similarly, there is enhancer insertion as well. Enhancer is another region in the genome of the virus that can also result into enhanced production of messenger RNA, resulting into the production of some specific proteins like growth proteins. So the insertion of viral enhancer into the host genome can also result into the activation of MYC gene, and as a result of which there is cellular proliferation leading to cancer. The fourth most important mechanism is chromosomal translocation, which is seen quite frequently in cancer cells. In this mechanism, there is translocation of one piece of chromosome to another, and it may be involved in the pathogenesis of Burkitt's lymphoma, which is the cancer associated with B cells, and in the pathogenesis of Philadelphia chromosome, which is the genetic abnormality associated with chromosome number 22. Chromosomal translocation also results into activation of MYC gene, which may induce cellular proliferation, ultimately leading to cancer. Fifth mechanism is gene amplification, which is seen quite commonly in cancers. It may result into the production of multiple copies of oncogenes, which may result into increased production of growth promoting proteins, ultimately resulting into cancer development. Now we are going to see in detail tumor suppressor genes. Tumor suppressor genes are those genes which suppress the growth of tumors. Basically, they produce a special protein that may inhibit cell growth or cell division. Tumor suppressor genes are also called cancer suppressor genes, growth suppressor genes, and anti-oncogenes. There are two main functions associated with tumor suppressor genes. One is gatekeeper functions, another is caretaker functions. By gatekeeper functions, tumor suppressor genes monitor cell proliferation. They control the rate of cell proliferation by monitoring cell cycle and apoptosis. Their caretaker functions include that they can preserve or protect the integrity of the genetic material or genome by correcting any damage which is imparted to nucleic acid material. And they can also maintain the stability of chromosomes during cell multiplication or cell division. But when tumor suppressor genes themselves undergo mutation or alteration, then they cannot control the growth of cells, and as a result of which there is increased cellular growth or cell multiplication or division, which may ultimately lead to cancer development. Here are some oncogenes and tumor suppressor genes enlisted, and we have already studied their functions and their association with different types of cancers. So MYC is a type of oncogene which is seen mutated in many types of tumors or cancers. It mainly affects cellular growth, then progression of cell cycle and replication of DNA. Then we have tumor suppressor gene P53. P53 normally involved in cell cycle arrest or in programmed cell death. This is also involved in biological aging, which is also called as senescence. Then it is also involved in DNA repair. This is also considered as regulator of cellular metabolism or the guardian of the genome, but it has been seen mutated in around 50% of human tumors. Then we have oncogene RAS. This RAS gene is available in many forms, for example, as KRAS, as HRAS, or as NRAS. This basically encodes a small GTPases, and these small GTPases may result into increased stimulation of adenylyl cyclase, and as a result of which signal transduction cannot be stopped, and hence it may contribute to the development of many types of cancers. Then we have RB. RB is a type of tumor suppressor gene. This is the regulator of the cell cycle. But if this tumor suppressor gene is mutated, then it may result into development of retinoblastoma, which is a type of eye cancer, and this has also been seen to be involved in the pathogenesis of some other types of tumors and cancers as well. Okay, students, activity for you. You are supposed to recall today's lecture and give the answers of these two queries. I'm going to reveal the answers. If you have answered both queries appropriately, it means you have understood the all concepts of today's lecture.